and I'm from group 9. Today our topic is muscle contraction. There are 3 types of muscle in our body: skeletal muscle, smooth muscle, and cardiac muscle. The skeletal muscle is a single nucleated muscle which is made up of microfibers. The microfibers are made up of myofilaments, which is further divided into actin and myosin filaments, which contain troponin, tropomyosin. They appear in a straighted shape and they are regulated by the nervous system. The smooth muscle of our body is a spindle-shaped muscle which does not contain the sarcomere, the sarcoplasmic reticulum, T-tubules, troponin, actin, or tropomyosin. It is regulated by the hormones and cell excitability and neurons. The third and last type of muscle in our body is the cardiac muscle, which is an involuntary muscle, and it is a straighted muscle which contains the single nucleus, and it contains the sarcomere, actin, myosin, troponin, and T-tubules. It is regulated by the nervous system, and it is self-excitable muscle. Today our topic is about the contraction of the skeletal muscle. So let's get started.
Action production, I stimulate the muscle contraction.
We are Z lines.
The space between the two Z lines are called as sarcomeres, and the sarcomeres contain I band, A band, and the third one H zone. During the muscle contraction, the sarcomeres contract and shorten with the I band, and the A band and the H zone remain still constant.
We are actin,
and we contain troponin and tropomyosin, and we bind to myosin and form cross bridges.
We are myosin. We have 2 heads, one bind with actin and other bind with ATP.
During contraction, we form a cross bridge with action.
I am tropomyosin, and I cover the binding sites of myosin on actin filaments.
I am troponin. I regulated the muscle contraction by binding with Ca+2, and I have three types: troponin I, troponin T, and troponin C.
I am troponin T. I have greater affinity to bind with tropomyosin.
I am troponin C. I have greater affinity to bind with calcium ion.
Calcium ion, I am bind with troponin C, so then actin and myosin could form cross bridge.
I am ATP. I bind with myosin so that their cross bridges can break.
At the neuromuscular junction, the depolarization caused the action potential to generate. This action potential travels. This was called the calcium ions in the sarcoplasmic reticulum to release. The calcium ions would bind to the troponin C on the actin filament. This would change the troponin tropomyosin complex and would cause the binding site on actin to expose. The myosin head now can attach to the actin filament. The cross bridge is formed, and this causes the I band to shorten. To break this cross bridge, ATP attached to the myosin head. The cross bridge break and muscle again get to the relaxed position. Troponin tropomyosin complex is reformed and muscle gets in a relaxed position. After this, the calcium ions would travel back to the sarcoplasmic reticulum in the sarcolemma. Calcium ions are stored here until the next action potential arrives.